Hello, my name is Petko Ivanov. I'm the Biosafety Specialist for Rice University. Welcome to the annual refresher of biosafety training. Some factors that complicate our task of keeping personnel safe in a biological laboratory are working with unknown samples and unknown organisms, working with higher concentrations and larger volumes that we might find in nature, and working with equipment that can generate aerosols. Any discussion about biological safety must include a working definition for biohazardous materials. Biohazards are defined as materials of a biological origin that have the potential to produce harmful effects in human beings. When we talk about biohazards, we generally think of viruses and bacteria. However, we should also consider more exotic organisms such as fungi, animals, and protists. Additionally, there are materials that are not living organisms, such as allergens and toxins, which are also considered biohazardous. Biohazards are generally denoted with the biohazard symbol. Human etiological agents are divided into 4 risk groups, depending on the type of disease they cause, how severe that disease is, and whether or not there are therapeutic agents available. Risk group 1 organisms are not associated with disease in healthy human adults. Risk group 2 organisms are associated with disease, but the disease is rarely deadly and there are therapeutic interventions available. Risk group 3 organisms cause disease that is more often fatal, but there are therapeutic interventions available, and risk group 4 organisms cause deadly disease for which there are no medical interventions available. While risk group tell us what kind of risk is associated with a particular organism, containment is the set of practices we employ to both protect ourselves and prevent the release of materials into the environment. Primary containment is the set of practices we employ to protect ourselves in our immediate work area. Secondary containment is the set of practices we employ to prevent release of materials into the environment. While recombinant, synthetic, and naturally occurring DNA is never a biohazard, it should still be handled with biological containment. Here's a list of the standards for working in a biosafety level 1 laboratory. This includes no eating or drinking in a laboratory, no mouth pipetting, and decontaminating your work surfaces throughout the day. Biosafety level 2 containment is appropriate for working with biohazardous materials. This may include materials and pathogens which infect us through the oral route, through breaks in our skin, as well as bloodborne pathogens. The standards for working in a biosafety level 2 laboratory are very similar to and build upon biosafety level 1 standards. There is an emphasis on personal protective equipment and sharps because we're working with biohazards. Additionally, there is a requirement for the employment of a biosafety manual and delineating the presence of biohazards with the biohazard symbol. So what is the appropriate dress code for a biological laboratory? Whether you're working with biohazards or not, the dress code is the same. You need to have closed-toed shoes and long pants while working in the laboratory. When working in a biosafety level 1 laboratory, you're only required to wear gloves, safety glasses, and a lab coat when handling hazardous materials. When working in a biosafety level 2 laboratory, where biohazards are present at all times, one should wear gloves, safety glasses, and a lab coat at all times. Though biosafety level 3 and 4 laboratories exist in the world, there are no biosafety level 3 or 4 laboratories at Rice University. There are a number of safety committees which apply to research involving biological materials. The IBC, or Institutional Biosafety Committee, conducts a generalized risk assessment of the type of work happening in every laboratory. The IACUC, or International Animal Care and Use Committee, oversees the responsible use of animals in research. And the IRB, or Institutional Review Board, oversees the responsible use of human subjects in research.